So I'm with Translatum Medicus Inc, and we're focused on macrophage modulation in blinding eye disease. Now, even though we're preclinical, my lawyers like it when we put this in, so these are our forward-looking statements. Now, everyone in this room, I'm sure, is aware of the unmet need for dry, age-related macular degeneration, and of course, latent disease, patches of tissue die away, and they relentlessly expand over time, and there's no treatment. Part of the reason we have no treatment for this unmet need is clearly the disease is complex, it's age-related, it's multigenic, and it's a disease of the innate immune system. And most of the therapeutic trials to date have focused on complement, which is the non-cellular arm. The second problem is clinical trial design is difficult, and we are currently measuring the rates of patch expansion, which is after the horse has bolted, and sustained delivery will be mandatory. But we're very different. We focus instead on macrophage modulation, and this is the cellular arm of the innate immune system. And our hypothesis is that resting macrophages become activated in the eye and then gobble up the tissue, because you really have to ask the question, who ate it? And so we believe that if we can convert an aggressive macrophage back to neutral, that we can both prevent geographic atrophy and prevent its expansion. And that's our therapeutic approach. So our rationale for embarking on this, because the scientific literature sort of led us away from this for the long time because of abnormalities within mutant mouse lines. But it turns out if you look in human samples, you can see macrophages and markers of polarization in the tissue. And when you do, there's a wonderful paper looking that shows some of these markers are only seen in late disease. So our science focuses on the whole transcriptional network within these macrophage, because macrophage behavior is obviously complex, and you can describe it within the macrophage transcriptome, with aggressively phagocytic macrophages being known as M1. It turns out that Mother Nature gave us a switch, and during non-inflammatory conditions, you get very low levels of transcription of some of the key regulators of the M1 response. During inflammation, you get increased transcription off the enhancer promoter sequences of some of the key genes, such as MCP1, 2, and 3. And our lead drug candidate, TMI-18, essentially targets this switch and prevents the transcription factors from leading to this increased transcription. So what we know from our preclinical models is that when we apply TMI-18 to cells in culture or in vivo, we can see that we modulate the M1 response, and we don't modulate the M2 response or the background M0. We reduce macrophage infiltration into the tissue, and in so doing, we actually protect the RPE. In a model we developed in-house, we can see that with TMI-18, we can dose-dependently block patch formation, and those blue patches there, these round circles show pictures inside the rodent eye, and in the presence of saline, that blue patch is very large, and as we increase the intravitreal amounts of TMI-18, we can effectively reduce that size until we can completely block it. Further, in a model where we use inflammation to create expansion, just like the patients in phase 3 trial, we can completely block patch expansion, and we've now demonstrated this in 2 species. Further, because this is affecting an entire transcriptome, we have a significant effect on downstream inflammatory mediators such as the inflammasome. We also modulate complement, and we have now shown that in our own non-GLP safety studies that this compound is entirely safe in the eye. So outside of our science, we've been very busy, and again, we are a very early company. We've had our first angel round. And already we've got an international patent portfolio, and I will say that I personally own all the IP. I wrestled it away from my hospital, and so it is now being assigned to TMI. We've got patents around the world, and we are now extending our indications and our related research. So we've also initiated our API production, and this was a big step in the last few months. And with that new GLP material, GMP material, we'll be able to perform our GLP tox studies. And we already have some in vitro data that shows that our small, low molecular weight synthetic is suitable for extended release, and that this can be measured in months. And the nice thing about working with a low molecular weight synthetic versus a biologic, of course, is that they don't denature over time. And we've confirmed that what comes out of the material months later is the same as what we put in. We've already initiated, we are just about to initiate our formal dialogue with the US FDA through consulting CMO with global experience. And this is our team. I think most of you know I'm a retina specialist and founder of this company, and I have a background at Novartis. Ken Howling is our CFO, and Natalie Pankova is my ever-so-smart PhD student that I one time here announced had just finished. She's now my Director of Operations. She's moved on with me. And then Dr. Keith Gordon. And many of you know these are some of our advisors here, and we're thrilled to have them on board. And this is our talent, and I point this out because TMI was selected to be a member of the JLabs at Toronto facility. And you may also know we started a second company called Tracery, and that was also selected, so we've got 2 companies within the Johnson & Johnson incubator. So this is probably why my lawyers put in the forward-looking statement. You can see that big arrow going up on the right. But what we've got so far, we had our first angel round 18 months ago, and we are currently looking at our costs and hoping to move forward. And you can see in this drug development plan, what we'd like to do is get to clinic very rapidly with our immediate release formulation, and then follow on thereafter with our extended release, and in fact push forward into phase 2 with the extended release. So our value proposition is that we have a first-in-class macrophage modulator for the treatment of blinding eye disease, and with a patented lead drug candidate suitable for sustained delivery, and we plan to move into AMD either alone and or in combination therapy. As you can imagine, being a macrophage modulator, we're very well poised to work along with the complement inhibitors. So thank you very kindly.